Hi students, today we are discussing few important concepts in the second chapter sexual reproduction in flowering plants. The first concept today we are going to learn it is about advantages of seeds. Before going to study the advantages of seed, we should know what is a seed. So we know that seeds are miniature plants. They are fertilized ovules which are used by the plants to continue their generations. Now, when it comes to the advantages of production of seeds or seeds produced by the angiosperms, here are some advantages I have listed below. So let us discuss those advantages. The first advantage, the produced seeds have better adaptation for seed dispersal mechanism. So what is a seed dispersal mechanism? Seed dispersal mechanism is nothing but spreading seeds from one area to another area or dispersing seeds from one area to another area. So it is very important in order to maintain their vegetation without competition. If all seeds they fall on same area, if this seedling starts in the same area, they start to compete for the nutrition for their development and growth. In order to reduce that competition, the seeds has to be distributed from one area to another area. They has to be dispersed from one area to another area. Then second advantage, they have reserved food material. So we know that during the double fertilization, the second kind of fertilization, it is triple fusion. So due to the triple fusion, the formed product, it is endosperm. Okay, so that endosperm is nothing but reserved food material which store the food material in the form of starch. So we know that the seeds don't have any greenish structure for photosynthesis purpose. So while they are germinating, while they are seedling, they don't have photosynthetic parts, but they need the energy for their development, their growth. So in such movement, in such condition, without photosynthesis, they need to have energy, and it is supplied with the help of the reserved food material. And third one, seeds have dormancy period. We know that if the seeds they were able to produce seedlings immediately after the formation of seed, you could have not stored them. So it could have not possible to store. So because of dormancy period, resting period of seed, so we can store them at our houses for our agricultural purposes, future agricultural purposes. And fourth one, we know that the seed is nothing but the fertilized ovule. So integuments only convert into seed coats, and the function of seed coat is to protect embryo, which is the actual and living seed. So in terms of biology, okay, in terms of agriculture, okay, then seeds are the basis for agriculture. We know that we may have irrigation, we may have fertilizer. Without seed, they are useless. So seeds have become the basis for agriculture. And the last one, seed viability. Okay, and the seeds have longer viability too. Okay, so when it comes to the seeds viability in the sense it is an ability of that seed for a certain period. So when we discuss about this, different kinds of plants have different kinds of seed viability. I mean, some plants may be viable for only 1 month, some of them they may be for 6 months, or some of them they may be for a year, and some of them they for thousands of years also. Okay, here I have mentioned two examples. The first one, it is Lupinus arcticus, where the seeds can be viable for up to 10,000 years. And the second one, it is Phoenis dactylifera, so date palm plant, so which can be survive, okay, which the viable can be the seeds can be viable for about 2 years. Okay, then next concept today we are going to learn, it is apomixis and polyembryony. So here in the first two cases we can find out that in both cases, ovule have been developed into seeds only. But the difference is, in one case there is fertilization, in another case there is no fertilization. So if the ovule get developed into seed after fertilization, it is referred as a sexual reproduction. In the second case, the same ovule had been developed into seed, but it is without fertilization. So that mechanism, that phenomenon, that process is called as apomixis. So apomixis can be defined as the development of seed without fertilization. Okay, here well-known examples for the apomixis are Asteraceae members. So Asteraceae members include Tridax, so as well as the sunflower, and the second one is Poaceae family, so where the grasses are included. In both Asteraceae and Poaceae family, most of the cases we can find the apomixis process. And when it comes to the types, there are three types we are going to discuss here: parthenogenesis, apogamy, adventitive type. So when it comes to the first one, apomixis type, that is parthenogenesis. So here parthenogenesis in the sense the development of embryo. So note that I have already mentioned that technically actual seed is embryo. So I am using again and again embryo. Okay. Now when it comes to the parthenogenesis, so we know that parthenogenesis is nothing but the development of organisms from the egg without fertilization. In the same way here also, if the egg developed into a embryo, a seed without fertilization, then we are going to refer it as a parthenogenesis. In this diagram we can clearly find out that here the egg has been developed into embryo. Okay, so that embryo may be haploid or embryo may be diploid based on whether the egg is diploid or haploid. So usually we know that eggs will be haploid in case if meiosis had occurred. If not, the embryo, the egg which is going to be formed, it will be diploid in condition only. So in certain cases, during the formation of embryo sac, it might have skipped the process called as meiosis, so that the formed embryo sac will be diploid in condition and all the cells present in embryo sac are diploid in condition, so that in such cases even if egg develop into a embryo, it will be in diploid condition. Otherwise, if the embryo sac is haploid in nature, then the egg will also be haploid in nature. So in such cases the developed embryo can be haploid in nature. Then second case, it is apogamy. So apogamy in the sense, apart from gamete, embryo sac has antipodals and so the synergid cells. If the embryo had been developed from those two kinds of cells, either antipodals or synergids, then the condition is called as apogamy. So note that if the embryo had developed or else antipodals or synergids contribute in the development of embryo, then the apomixis is called as apogamy. And next type is, it is adventitive type. So when it comes to the adventitive type, we know that apart from embryo sac cells, it may be nucellus cells or it may be integument cells. If they contribute in the development of embryo, then we are going to call it as a adventitive type. We know that in megasporangium there are two protective layers called as outer and inner integuments, and inner integuments there is an iterative tissue called as nucellus, and inner to nucellus there is embryo sac. If the embryo sac is not involved, only either nucellus or integument is involved in the development of embryo, then we are going to say, then we are going to call it as a adventitive type. In this adventitive type, so it may leads to the polyembryony. So then what is polyembryony? The name itself indicates poly means many. Here in case most the seed development, we will find that in a seed there will be only one embryo. In certain cases, instead of one embryo, the seed may possess more than one, it may be two or more than two. In such cases we are going to call the condition as polyembryony. So usually in case of adventitive or adventitious apomixis type, usually the polyembryonic cases can be seen. So once again I am mentioning that polyembryony means it is a condition in which seeds have more than two embryos. So example, citrus and mango.